Okay. Okay, we're very lucky to have Jose Luis Ambite, who is a professor in computer science and also at ISI, both here, here at USC. Expertise in a lot of things, but you're going to talk to us today about genetics. So, and then also Ruhi Shemirani, and he's a postdoctoral fellow at the Institute of Genomic Health at Mount Sinai. So without further ado, here they are.
Yeah, thank you.
Thank you.
So we're going to talk about IBD mapping, or step-by-step mapping, and how to make it at a scale. But before I start that, I should say how we came to this place. So I'm a computer scientist, and Ruhi is, so he's more of a computational geneticist now. And but I have interest in genetics, and I'm part of a large genetic study called PAGE, Population Architecture for Genomics and Epidemiology, which is a genetic study of multiple populations. And I was sitting in one meeting, and I learned about this problem of IBD, identity by descent, which is to find if you share some genetics with somebody else through a common ancestor. And as I was listening to that, I realized that the algorithms that existed were not very efficient. And I thought, based on other background that I have from data integration and record linkage, that I knew how to solve that problem. And I had an idea. And I proposed this to Ruhi as a summer project. And then 3 years later, I guess, or so, it was his thesis in computer science. And now we have one of, well, probably the most scalable algorithm to compute this problem, identity by descent. And that led to a whole pipeline on identity by descent mapping. And I guess the moral I hope is that you should talk to your computer science friends that sometimes may have some ideas of how to improve your processes. And hopefully you have that idea. We'll talk after the presentation. So we're going to briefly cover a bit what genetic association studies and what IBD mapping is, and how we, with different steps, which IBD estimation, which is the first step, clustering and statistical analysis, and how it was Ruhi validated these results. So this goes into the overall umbrella of genetic science, which is understanding, predicting human traits and diseases based on genetic data. And now it's a big data science problem, as you know, that very large genetic data sets available, that becoming larger and larger. In the study in which we started, it was reasonably large. It's a consortium of different cohorts, and it has about 50,000 participants. But you all know that Biobank with 500,000 people, great resource. Kaiser is maybe a bit updated, probably have more now. The million metadata programs with 1 million, all of us 1 million. China, Genome-Bank also 1 million, and more are coming. There are all these biobanks with lots of genetic data. Actually, the UK Biobank just released 200,000 full genomes, I think. So the idea of a genetic association study is that most diseases are a combination of the genotype, the genetics, and the environment. And there will be some kind of genetic marker that you can measure and mix it with some other phenotypic variables, covariates that measure the environment, and that help you predict some disease like cancer or type 2 diabetes and so on. As you know, the human genome has like 3 billion pairs, while the duploids, so like 6 billion base pairs. There are different types of, you can do whole genome sequencing, you can measure the 6 billion pairs. Oh, maybe can somebody mute? Otherwise, the whole exome is you only capture the part of the genome that code for protein and kind of the surrounding areas that generally have enhances, promoters, regulatory regions. And then genotyping is only captures, say, like 1 million, 2 million points. That are particularly interesting points. And these are called single nucleotide polymorphisms, that are points of common variation in human populations. And then you can analyze if any of these SNPs correlates with some disease. Of course, genotyping is very cheap now. I know it's order of tens of dollars. Whole exome sequences are becoming cheaper, and whole genome sequences is becoming cheaper every time. most of what we talk about applies to genotyping, although we think all the base techniques will also apply to sequence data. So for a single nucleotide polymorphism, as I was saying, it's a common variant, generally defined as having a minor allele frequency of more than 5%, or yeah, so it's not super rare, it exists in a population. And it is used because it represents like other more rare variants. It kind of tags the causal variants, the ones that actually cause the disease, although imperfectly. So the genome association study, you get some population, they have some cases, some controls. You have, say, 1 million SNPs, maybe you also impute them, so maybe you have 50 million SNPs, and then you run 50 million regressions for each disease, or each trait that you are interested in, and you will get a plot like this that many of the correcting for multiple testing. There will be some hits somewhere that exhibit a strong association between some part of the genome, some SNP, some set of SNPs, hopefully, that signals association with a particular trait or disease. But these SNPs that you find are not the causal variants. The SNPs are kind of correlated or associated with the causal variants. Again, this is an example of distribution of the allele frequency. So you see that depending on the frequency, there will be most of the variants are these kind of very rare variants, and it has kind of a long tail of the more common variants. But these rare variants can have impact of human diseases. And the reason we're talking about rare variants is because IBD is a way to, or this IBD mapping is an approach to get a handle of rare variants without having to do the full sequencing. So if you have the full genome sequencing, then you can get the variants, read them directly. But IBD is a way of getting to them, and it's a way of doing a family study, but in kind of sense, very distant families. So what is IBD? IBD, as I said, is do you share a little segment of DNA with another person? And generally, if the segment is long enough, it's because you have a common ancestor. And that's what we call identity by design. So here you see a tree of people with the 2 chromosomes, and as they kind of mix, they marry, they have children, you will see that eventually maybe these 2 pair of people share a little segment that came from one ancestor, which is this kind of grandparent or grandfather. And these SNPs, even if you find them through just SNPs, they are probably close and tagged in a region, the rare variants that maybe cause some disease. So an IBD haplotype is this segment of DNA inherited from a common ancestor. The length of the shared IBD segments actually kind of decreases with how many generations that common ancestor lives, and it's kind of you see this 1 to the n, the number of generations. So IBD is really exploring like kind of recent common ancestors. But you can do this kind of, you can take these people that you're related to through a common ancestor and think of them as a little family. And in a sense, you can do a family kind of as a study over a large data set. So from the computer science point of view, which is like the first time I read this, is very simple. Is you find some matchy substrings, some segments of genetic data longer than some given length. So we want to be long enough that we know is IBD. And you want to find that quickly among all pairs of samples. And here you have some representation of that in which these are the SNPs that you measure, no? It's kind of SNP 1, SNP 3. And these 3 people, these 3 individuals actually share this, say, string of n SNPs in a row, and that can only happen if you have a common ancestor. It would be very rare to, if the string is long enough, it would be very low probability that happened by chance. But what happens is that this segment has some kind of rare variants and is kind of representative for those rare variants. So this little group here is like a little family, distant family, very distant family, because it's a very small segment. But it can help you to get a handle on these rare variants. So discover if they are associated with some disease. So if again, maybe this group of people have a higher prevalence of a disease, then you would say that you would discover that through this IBD mapping process. So we're going to describe the IBD mapping process and scalable methods algorithms to do this. So we didn't embed the IBD mapping approach, but we develop a number of methods to make it scalable. Since we have this many biobanks. Okay, so this is like a conceptual overview of the process. So you have a number of people here, this kind of six kind of haplotypes, and there's some real IBD that is shared. They got from some common ancestor, and let's say that through kind of the complication of genetics, these kind of 4 people, numbers 1, 2, 4, and 6, they actually share these kind of golden areas from some common ancestor, and then these other 2 people, 3 and 5, they share the other region from a different common ancestor. So you could kind of break this genome in windows, and we'll let you know later how to do those windows. And then you can see, let's focus here in window N, and window N+1. So we can see, okay, who does the algorithm? We have an algorithm that is able to tell us, okay, in individual 1 and individual 2, actually, these segments are kind of identical or very, very close. So you would create a little graph that said, well, 1 and 2 share a little segment in this specific window. And then maybe in this next window, they also share a little segment, but they're farther apart, they don't share a segment. So you do that across the genome, and you create these little clusters. So first you estimate who shares with whom, and then you're going to cluster them. And you see here that this looks like almost a complete click, but not quite. For some reason, we didn't discover that 4 and 6 actually were share the genetic data. There is some flaw in the algorithm or some other complicated problem like phasing. But we can discover that since like sharing a segment, the kind of equality is basically is the segment equal to be transitive, then these little clusters should be cliques. So we can essentially complete the cliques, and then we can figure out, should it be a complete click? So here maybe this should be a complete click. So this missing link here was a missing false negative, or here maybe there was a false positive. Maybe actually 2 and 6 didn't really share genetics in a share segment in reality. So the click should be smaller. This is the 1 to 3. And this is another computational problem that, as we see, is not cheap to do. And eventually, what you do is this idea of, well, now we have these little groups of people, these little clusters. I'm going to call them, using notation like a distant family, very distant because these segments are small. But if now in this particular segment, there's some disease that kind of this segment is enriched for some disease that is much more prevalent in the cluster than in the general population, that can tell you that in this particular region, they could be the genetic cause of that disease, the genetic basis for that disease. And that's kind of the idea of the IBD mapping. And you have to develop now what are the right statistical methods to make sure that this kind of hypothesis test of, is this really a valid association? and that's where I think we can get lots of help from this community. So again, so here's the same idea of the kind of the IBD mapping.
Jose Luis, there's a question in the in-person audience.
Okay.
On the previous slide, so these clusters, each link represents a shared subsequence.
Yeah, each link is literally sharing a little segment of DNA. I mean, it's the SNPs. So, yeah. But based on the SNPs, we assume that the whole segment is shared, but it's kind of some rare variation encoded there. Yeah, so we literally find these substrings. Like for the computer science point of view, is like, do we share this substring?
But it doesn't have to be the same substring, or else it would always be you're not looking for the exact same substring, or else everything would be a fully connected link, right?
Sorry, I'm having a bit of a hard time hearing you, but
so it doesn't have to be the exact the exact match.
No, so we allow for some. So we have a degree of similarity in that segment, and we allow for not being. So we allow for having some small variation, but they tend to be like a string equal, so like the same substring. But we can set up the algorithm to allow for some small variation. In these cases, you literally are looking for a share segment from a common ancestor. So it is like the same string most of the cases, but you can assume that even some like random mutations happening, even if it's like a few generations back, and we can set up the algorithm to not be completely exact. Is that the? I mean, I'm sure there's an ulterior motive why you're asking this question. They might be clarified later in the talk, but is that are you satisfied with the answer, or do you want more detail?
I didn't understand if 1 and 2 are connected and 2 and 3 are connected, then wouldn't 1 and 3 always be connected if they're sharing the exact same, if they're sharing almost the same sequence?
No, so they should be connected. They should be connected. I mean, that was a conceptual representation of maybe the algorithm, for whatever reason, it didn't find them. Okay, but they should be connected. So that those little clusters should be cliques. They should also be connected if they truly share the segment.
Okay.
Okay. Oh, there's another question.
Oh, that was Abigail. So
Sorry.
Abigail.
thank you. I think we're good.
Oh, that was you. Sorry, I see the hand raised. Yeah, I should have maybe put the 2 monitors. It's just I'm doing 1 monitor, so it's hard to manage. Okay, so what we're going to do is like it's an alternative to typical GWAS methods, but now as opposed to have SNPs, we're going to have membership of clusters, and we still will run some kind of regression to figure out does being a member of a cluster associates with some trait or disease under with some covariates. So that's kind of the overall of the. And again, we didn't embed this method. This has been applied by some of our collaborators. Icahn, it was used to discover some association in a Puerto Rican population. Well, so this was another method for association. This one, they found a collagen disease in Puerto Ricans that came from some Native American ancestry in this kind of very related population. It's in Puerto Ricans. It was a kind of bottleneck. And they were able to find this disease by using IBD mapping that they couldn't just find by using traditional GWAS. And there's a number of other people that have actually worked in this idea of IBD mapping. So what's the challenge? The challenge is that conceptually it is very simple, but it's very complex to compute. It's computationally complex to compute these pairs. So there's a problem of scale. So it works fine if you're doing like, I don't know, 3,000 people or 10,000 people, but when you go to 50,000 or 500,000, then you don't have enough computers to run this with the existing algorithms. And then the issue of, well, how do you do these statistical tests? And that's actually underexplored, and Ruhi came with some solutions, and we welcome more ideas from the audience, actually. So I'm going to, I realize I'm a bit slow. I'm going to speed up a little bit. So the 3 steps of IBD mapping is IBD estimation, clustering, testing. So we'll go to the first algorithm, which is the one that, okay, we can find these common substrings, and it's really kind of string equal. That's the easy way to think it. And you think it's actually a very simple algorithm. You could just take all pairs of individuals in population and just scan the genome, and you will find all the subsegments. That would be a very trivial algorithm. The only problem with the algorithm is like it's n squared. And when you have like 500 people, 1 million haplotypes, 1 million square is a really large number. And you have 1 million, so a trillion or 1 billion square times maybe 1 million SNPs, that will take a long time computationally. So we came up with essentially better methods. But for the computer science problem, is a very simple, well-defined problem, is find all the maximal segments longer than a given length that are shared between any substrings of these long strings, the genome, in a large set of strings. And we can allow for a few mismatches, but often we find exact. So there's a number of, so how do people have solved this problem before? They do hashing, this will be the first thing that you do, and we just came up with a better hashing algorithm, essentially. The existing kind of standard, at least when we started this work, was germline, and that actually, they had run it on page before we came up with the idea. And then when I learned that actually it took like 6 days per chromosome in the, I think, Mount Sinai's cluster, they run every chromosome in a different computer, but it still took about 6 days, which I thought it was like quite long. There are other more efficient methods that came later, like Rapid, that uses the Boros-Willis transform. Unfortunately, even it's quite fast, but it has low accuracy and recall, at least in non-experiments. And there's some other approach that called Refine IBD that uses germline, but a hidden Markov model to really find the IBD segments, and it's quite accurate, but it's slow. So our contribution in this algorithm called ILASH that uses locality sensitive hashing to find all these pairs quickly. And I'm going to skip some things because I think I won't left enough time. So how does everything work? So you take the whole genome. So look here at the picture. You have your own genome. You define size of interest, and it's not. I mean, you could do it on a length based on the SNPs, but since the density of SNPs changes, and what really matters in genetics is like this kind of a recombination rate. So we measure it in centimorgans. So you say that you want to find segments longer than 3 centimorgans share between 2 pairs of people. So you would take all your individuals and find out what are regions of 3 centimorgans. So you kind of create windows of 3 centimorgans. And in some cases, those windows will be kind of in true base pairs and true genetic distance will be like bigger regions or smaller regions, depending on the density and the recombination rate. But you split all the genome in these slices. And then you're going to take each slice, or you're going to, well, actually you take first the whole genome and you create little hashes, or maybe say 20 SNPs. So there's little hashes. And then you're going to split all those hashes into these windows or slices of, say, 3 centimorgans. And depending, I mean, in some area, it could be 300 SNPs or 500 SNPs. In some areas, maybe only 80 SNPs. And then you're going to compute this thing that you call Minhash, that is something like of a magic algorithm, but what it does, it computes the GWAS similarity between two segments. So the big idea is we're going to take the genome, we're going to split in windows. Within window, we're going to split each the genome in little segments of, let's say, 20 SNPs, 20 bits, and we consider like major allele, minor allele, so it's like 20 bits really, there are only 2 values. So you have these like 20 bit segments that you split inside here, and you are going to do something, it's kind of crazy at the beginning, but it actually works quite well. So you're going to turn this little segment into a set of hashes, each of 20 bits long. And as I was saying, in some segments that may be, I don't know, 50 hashes, in other segments will be more or maybe less. If you have very few hashes, the algorithm will have less precision. So you've taken a problem from a linear sequence match into basically matching sets. And what we're doing is do these 2 sets of 20, I mean, I say 50 hashes, 50 elements, how many elements in common do they have? And a way of measuring those elements common is called the GWAS similarity. And it's just now there's an algorithm that can compute the GWAS similarity among very, very large data sets very, very quickly. And I'm going to tell you briefly how it works. And you'll have to trust me that this actually does what it's supposed to do. So you take an individual, and now an individual in a particular window is composed of a set of hashes. It's a set of hashes. Hashes 1, 2, 3, 4, 5. Okay, these little 20-bit segments. And it said that individual 1 has the little hash of type 1, and the of type 3, and of type 5. Individual 2 has 2, has 4. Individual 3 has 1, has 2, has 4, and so on. And you have your million people arranged this way. And then you're going to compute some kind of permutation of the hashes. You'd say, okay, I'm going to check hashes in this order, like first 1, then 4, then 2, 5, and 3. That's permutation 1. Then permutation 2 is H3, 3, 5, H1, and so on. And you're going to construct this matrix called the Minhash signature matrix. That essentially you look at the order of the hashes and you see in this order what's the first time that you see a non-zero. So you go for individual 1, in permutation 1, the first one you have to check is H1, and you see that H1 actually that individual has that hash. So you put a 1 here. And then for individual 2, you check it 1, it doesn't have it. You check H4, it has it. So it's the second time. So you put here 2. Okay, and then for individual 1, H1, it has H1, you put a 1. And you do that for every permutation. So here you look at individual 2 for permutation 2. Permutation 2 has an Does it have an H3 first? No, it doesn't. Does it have H5? No, it doesn't. H1? No, it doesn't. Oh, it has H4. Yes, it has. So it has a 4. Okay, so this is like an obscure procedure, but it turns out that once you compute this Minhash signature matrix, if now you see for each individual what their signature is, and you compare them, you compare how many values in common they are, that actually approximates the GWAS similarity. That computes the GWAS similarity. And it's a bit complicated, but it's not super complicated, but it takes longer to explain than we have in this talk, but that computes the GWAS similarity. So it tells you, and then you can implement this very, very quickly. And it will tell you, for example, here that it will help you find, the best thing of this is it helps you find who does not have anything common. So you can remove lots of possible candidates. So I said the worst case would be n squared. So you can remove the vast majority of the n squared candidates very, very quickly. And that's where you get lots of speed up in this algorithm. So here you see that, well, we actually found that I1 and I2 don't share any I1 and I2 don't share any, the signatures don't have any values in common. So they're not going to have a GWAS similarity. So these people wouldn't have to consider or have to compare them in detail. And then there's depending on how many hashes you use, you have a better approximation of this GWAS similarity. Okay, so now when you have a Minhash signature matrix, and this could be maybe 20, it doesn't have to be a very large number, but let's say 20 hashes or 20 permutations. So the bits of length 20, vectors of length 20, or 30 or something like that. Then you can split them in little groups and do a traditional hashing technique, and that very quickly find who is going to have min hashes in common, and again compute the probability. After, I guess, this approach is done, it turns out that the probability of having at least one LSH value in common after development of hashing has this probability being R is the number of values per band, the number of bands, and the level of similarity that you want. And this function has this kind of sigmoid type of shape, meaning that you're going to very quickly tell that some people are not related at all, and they won't match, or people are going to be related at all, and very, very related. And you can control kind of the curve by changing this number of bands, the number of hashes. And this was kind of the idea. This has been used in other domains to detect similarity of text and plagiarism. It's a non-algorithm in other domains, but it has not been applied to genetics. If you want to get a longer explanation of this algorithm, a better explanation, you can look at this book. There's a whole chapter dedicated to this algorithm. It's the Ullman book, Mining of Massive Datasets. So once we have all these candidates that we have with high confidence that they're either going to share a segment or don't share a segment, and the greatest value is eliminating lots of candidates that don't, we are pretty confident they're not going to share anything. Then the algorithm that Ruhi implemented, then it checks carefully the hashes, and it tries to not concatenate windows. So instead we slice the genome in different windows. So now we're going to see the results across multiple windows, multiple slices, and kind of extend that maximal segment. So because even we are finding the segments per slice through the LSH, it could be that the segment is a longer segment than 3 centimorgans, maybe it's 20 centimorgans. So we have to stitch together all the different, all the consecutive windows. This algorithm, I mean, that's definitely the core idea of the algorithm. There are also a lot of kind of implementation improvements using parallelism and interleaving, kind of reading the file with calculating things like using I/O time. So Ruhi, which is the main developer of the algorithm, did an awesome job on that front. But there's a core algorithm and then a lot of improvements and implementation. So let me quickly go through some of the results for the IBD identification. So it turns out that this method is actually quite accurate. It's comparable or better than existing algorithms. So this is an experiment on the recall. So how much of the ground truth? So we simulated data using the same kind of IBD distribution that we found in different page populations. So we have a population, is a multi-ethnic page, is a multi-ethnic study, and has Puerto Ricans, has African Americans, Asians, and so on. So we look at every participants in page, and then only Puerto Ricans and African Americans have different IBD patterns. And in all three cases, ILASH, which is our algorithm, essentially performs better than comparable existing algorithms, either Refine IBD and Germline. We also compare with this other new method that is very fast, but it has a much lower accuracy, so we're not going to compare more with it in this talk. We also look at what is the kind of accuracy of the system depending on the length of the track that we're looking. So we said that we're looking for IBD greater than a given length. So if it's greater than 20 centimorgans, essentially we cover 100% of the data. As it goes to smaller segments, the smaller that we tried was 3 centimorgans, we kind of recover about 80% of data with 90% accuracy. And that's kind of comparable or better than previous algorithms, except that in the very, very kind of high accuracy case, Refine IBD is a bit more accurate because they're using a more refined algorithm, this kind of clear market model to kind of find the details. But for more practical purposes, ILAS is generally better than previous work. It also has a very low false positive rate. So the way we tested this, or Ruhi tested it, is to generate like some genetic data that shouldn't have any IBD whatsoever. So you took data, split it in little 0.2 centimorgans, shuffled everything, so there should be no IBD, or just kind of random in there. And Germline, using this particular hashing, it found like 26,000 false positives. We found IBD like about 90, and we found 2. So we only had like 2 false positives. So it has a very, very low false positive rate, this algorithm. And now I wanted to put this slide first, but I wanted to tell you it was accurate before I told you it was fast. So it's actually much, much faster than previous algorithms. So this kind of blue line is page in slightly different populations. And as you see, these are synthetic experiments, up to 80,000 individuals. And I mean, Germline, it just kind of spikes quite quickly. It can handle, let's say, about 20, 30,000, but it really is like n square. Actually, all these algorithms have a kind of an n square behavior. The thing that the n square behavior in ILASH is very, very kind of slow. So it takes a lot longer to really get into the problems with n square. And again, so this is based on the number of individuals, and this is based on the length of the tracks found. So in a sense, our algorithm is almost linear on the number of tracks that are found. I think that the bigger the population, the more segments you're going to find in common. So this is like, we took 100 seconds for 80,000 individuals, while Germline was taking 18,000 seconds for 30,000 individuals. So this is kind of the improvements. Comparison with Rapid, so we're still also faster than Rapid, but the bigger problem with Rapid, I think Rapid can be done faster. Maybe there's new implementations that use the Borel-Wheeler transfer that are faster. The problem was that it was not very accurate. And then time memory growth, I mean, they're reasonable amounts of memory. So you can see here, 200 seconds in memory, I don't know, about gigabytes. It depends on how big this intensity it is. And the algorithm itself has a variety of knobs that you can turn, how many permutations, how many permutations per band, the Minhashes, and you will get different trade-offs of performance and accuracy. But if I mean, we can maybe take this offline. So we ran this on real data in Page, and then UK Biobank, and I think we were one of the first people to be able to run IBD on the UK Biobank with accuracy, or probably the first with accuracy actually. And we found, so there were almost 500,000 people, about 650,000 SNPs after QC, and we observed 12 billion pairwise connection. So in this group of 500,000 people, there are 12 billion kind of distantly related individuals, which is mind-blowing to me, it's about 10% of all the possible graphs, like the n-squared. And Gillian, who I think actually on the audience, used these IBD results to understand better or to confirm based on IBD what is known historically about the populations in the UK and their kind of Scandinavian kind of components. So that was the IBD estimation. There was kind of the initial algorithm that started this IBD mapping and scalable IBD mapping idea. And then Ruhi is going to take over and tell us about how to also efficiently do the clustering, because the challenge here is how can you do all this efficiently? So maybe Ruhi, I don't know if you want to present or you want me to present.
I can share my screen.
Okay. And I'm sorry, I think I'm a bit late. A bit late, and
I hope everyone can see my screen now. And can you hear me?
Yeah, we can hear you. We can see your screen.
Okay. So I realize that I'm short on time, so I'll be skipping over a bunch of slides. Feel free to just interrupt me with any questions that you have. So the next step, as Jose Luis talked about before, after extracting IBD out of the data set, is to create local IBD graphs by windowing genome into small sections. And while you would expect that everything that you see, every subcomponent that you see in these graphs to be fully connected and be basically clicks, in reality, we have false positives and false negatives due to various reasons, like phasing errors or the section being the IBD segment being too short to be showing up between two of the individuals, for example. These things happen, and thus we don't have the clicks that we would desire normally. Clustering algorithms are the solutions that have been in use to address this problem. We realize that not all of the clustering algorithm out there are appropriate for our purposes. So we did an analysis to find the most statistically powerful clustering algorithm for IBD clustering, for local IBD graph clustering. To do so, we had to create our own local IBD simulation benchmark because common graph benchmarks did not have, could not realistically represent local IBD graphs, as we will see in the next slide. So we simulated clicks, and we started by simulating clicks with realistic distribution and removing the false negatives and adding false positives for a range from 5 to 50% over 750 simulations. And for clicks that we simulated, for those that had more than 10 members, we also simulated binary phenotypes to basically be able to measure the statistical power of clustering algorithm that we were studying. As you can see here in panel A, you can see a local IBD graph in a real setting. On panel B, you can see the results of our benchmark. And in panel C, you see LFR, which is the most common benchmarking algorithm for graphs and clustering. We used, as I said, the binary phenotypes that we simulated to calculate a statistical power of various clustering algorithms in terms of how many of the simulated clusters they could, how many of the simulated phenotypes they could extract, and how much of the significance of these simulated phenotypes they could extract, because every cluster had a simulated phenotype that had a different prevalence inside the cluster compared to the entire population. And this difference in prevalence was statistically significant. So we not only wanted the clustering algorithm to find clusters that were enriched for a phenotype, but also the significance of that enrichment should be carried over. And that's how we defined our statistical power. We tested 5 different clustering algorithms. The first one was highly connected subgraph, which has been used for IBD mapping and other bioinformatic problems. And then we also picked Louvain and Leiden because they are not only really fast and they're scalable, but also they've been used in computer science and other problems with good results. We also analyzed InfoMap and Markov clustering algorithm because we knew that they have been previously used in bioinformatics applications. Specifically, MCL or Markov clustering algorithm provides the option to control the granularity of the resulting clusters. So we checked various four different values for the level of granularity, and we will be showing that. In general, we found that InfoMap and MCL with an inflation ratio of 1.5, MCL with inflation ratio of 2, and InfoMap have the best and most stable performance in different cluster counts and false positive, false negative rate scenarios. In terms of a speed, however, the worst performing algorithm before, which is Louvain and Leiden, had the highest speed by 2 orders of magnitude, followed by Markov clustering. And the original algorithm HCS, which was the commonly used algorithm for IBD mapping, was so slow that it couldn't quite finish all the experiments that we had planned. We found out that a phenomenon called resolution limit basically limits the ability of Louvain and Leiden and other greedy modularity optimization algorithm to perform well for IBD mapping, because they ended up lumping basically together and merging poorly connected clusters into the same cluster. This becomes more evident if you look at the average cluster size per experiment across our 750 simulation for Louvain and Leiden compared to the ground truth on the bottom, and see like how extended and wide the distribution of their cluster sizes is. We can also look at the average node connectivity and see that Louvain and Leiden, regardless of what we provided as in terms of false negative and false positive rates, they always ended up connecting as much clusters as they could in this click-based situation. We also looked at common clustering metrics to see how in the absence of a ground truth, we can use these methods, we can use these metrics to basically find out more about the performance of our clustering algorithm. And we found out that we cannot use them, and there is no realistic way of using any metric that is not based on a ground truth for our statistical power. However, we were able to find a use case for this. We found that they are informative in basically predicting and calculating the amount of false positives and false negatives in our realistic data sets. So we could use them to calculate the false positive and false negative rate of our realistic data set, and then create a benchmark that is realistic enough for our algorithms to basically find the most powerful algorithm on a database basis. We use this algorithm and the output of eyelash to find local IBD clusters over UK Biobank. As a simple verification of these clusters, we looked at rare variants in the whole exome sequence data of 200,000 individuals in the UK Biobank, and tried to basically find out for every rare variant if there is a cluster that covers the carriers of that rare variant. We found out that for doubletons and tripletons, we can fully recover 1/3 of these variants, while a null model, which is a randomized set of clusters, find close to 0%. For more common variants, for example, for those from 4 to 50, with 4 to 50 carriers of the minor allele, we found out that we can recover 1/3 of the variants with at least 25% recall, but the randomized cluster cannot do the same thing. These results were also later replicated by another more analytical IBD estimation experiment on UK Biobank called FastSMC. We packed our clustering solution with a simple association testing program into a software package called Icahn. And while Eyelash enabled a scalable IBD estimation, for example, it could do IBD estimation in page in under an hour, Icahn enabled a scalable IBD mapping by using Minerva supercomputer here at Icahn School of Medicine and Mount Sinai. It can analyze the whole of the UK Biobank in terms of clustering and linkage in under an hour, with 500 million clusters found. For every one of those clusters that we find, Icahn basically tries to test the associations between that cluster and various phenotypes. We currently support two simple approaches, one t-test. The first one is t-test, where we basically compare the average value of the phenotype in the cluster compared to the total population. And we also have added linear regression support, which corrects the phenotype value for age, gender, and population structure by including the first four principal components as covariates. And it then looks at the distribution of the effect size. We tested 4 different phenotypes: BMI, height, LDL cholesterol, and HDL cholesterol. We only tested clusters that had more than 10 members to deal with the inflation of our results, which basically filtered down our clusters from 500 million to 100 million. Here is a summary of our results. In total, we found 595 significant associations between these familial clusters and our 4 tested phenotypes. As expected, linear regression had a lower inflation factor in terms of the p-value of the results found compared to the t-test, while still delivering more significant results in a smaller cluster. One of the other predictable things was that for complex traits like BMI and height, the 2 methods found independent clusters, while for LDLC and HDLC, we had a significant amount of overlap. Here are a few samples of the results we found. I'm going to skip over them because I know we are short on time. Here's for height. I'll talk about it shortly. We have the same thing for LDL and LDLC and HDLC, and you can see the overlap between the results of the 2 methods. We tried to verify these experimental results using Genome Browser. So as you can see here, for example, with LDLC, we have the significant cluster. We look at the Genome Browser and the list of genes that are covered by the location of these cluster, and look for previous publications that associated these genes with our target phenotypes, like this. Here we had PLPP3 under the area of this cluster, and there was a previous publication about it. This way we could able to verify all of our results, but 2 for height, 1 on chromosome 9 and 1 on chromosome 15, which were not located in any area that were previously associated with height. Another interesting thing that we found was this paper that basically described 6 genes that could carry rare variants with high impact on HDL cholesterol. And for every one of those genes, we found at least one cluster, one family cluster around, or not around, exactly in the same area as that gene in our results, both using t-test and linear regression. We also compared our results to imputed GWAS results done by the new lab at MIT. They tested, their imputed data had 9 million points. Our data had 100 million points. And still our inflation was not as high as the inflation factor in imputed GWAS results. Again, for complex traits, we had little overlap, but for LDLC and HDLC, we had quite a bit of overlap. In terms of what happens with the clusters that we found, in previous studies, they use those clusters to identify actual rare variants. So for example, if we found cluster X that we found for HDLC with a p-value of 10 to the power of -65 and 36 unique members, we found that 17 out of 36 members had their whole exome sequence data available in UK Biobank. On chromosome 11, we found 1 million rare variants. We filtered down those variants to those in the same region and carried by the same family, which filtered those results down to 9 candidate rare variants. Here we are showing 4 of them, and the probability of a variant with that frequency happening in a cluster that small is shown here, and as you can see, it's pretty low. So the next step would be trying to look into these rare variants and their possible effect on HDLC. To summarize, we talked about ILASH, which does a scalable and fast IBD estimation in genotype datasets. And IBD estimation has many more downstream benefits and application other than IBD mapping that we talked about. And we also talked about a scalable clustering of local IBD graphs using Icahn, which will be published shortly, hopefully. For future work, I think the most important thing, if I want to mention just one thing, is finding more robust and well-calibrated statistical analysis tools to test these clusters for association with different phenotypes. And in the end, I want to thank Dr. Jinyu from University of Colorado, Drs. Belbin and Kenny from Icahn School of Medicine at Mount Sinai, which helped us develop these methods and guided us the whole way.
Great, thank you very much. Do we have any questions online or in the
I've got a question.
audience? We have a question?
I just wonder, how does the eyelash behave in the presence of admixture? So our populations.
Yes, were you able to hear the question?
Yeah, so I mean, eyelash is agnostic in a sense to admixture. So we tried in different populations that have different IBD patterns, but also different admixture, like Puerto Ricans are admixed. And so it is finding this kind of very small segments. So it's we're more like a local ancestry method really than anything that has to do with admixtures at a global level. And I don't know if that answers your question. I don't know if Ruhi, you have
I think in general, what we find, most of what we find has happened after admixture events. And also, as Jose Luis talked about, our reference data set that we developed eyelash for was PAGE, which is a highly admixed, which has highly admixed populations.
Yeah, we tried in Puerto Ricans and African Americans that are admixed populations. So,
I noticed you were correcting for principal components in the disease mapping. Is that right?
Yes. Yes, we are.
So just a follow-up question to your answer. Have you actually compared sort of local ancestry mosaic segments to the clustering sort of segments you get with the IBD to see whether how those overlap and maybe compare?
No, we have not, and that's actually that's a very good idea. That would be a very good next step.
Yeah, that might be interesting to see. To your point about capturing sort of more recent sort of relationships than local ancestry might. That would be interesting to compare.
Yeah, I guess like the local ancestry results would be further back historically compared to what we are finding, but it would be interesting to see the mosaic, as you said, on a single IBD segment probably.
Actually, we haven't really looked that much at local ancestry, but I know I suspect, or I would hypothesize that probably the type of hashing techniques that we're using, this local sensitive hashing, can also speed up algorithms to compute local ancestry. Yeah. So a lot of what we're doing is really kind of, it's not so much changing the, I mean, I think that many things that can be done on the statistical methods, but on the IBD is really making some of this IBD mapping scalable by be able to scale to large sets that you couldn't otherwise. And I think there are a number of algorithms that we could scale up using this type of techniques, including local ancestry, also probably phasing, but we haven't tried that.
Yeah. There's one other question in the audience.
So I have a question about the clustering algorithm is essentially the results of the clustering algorithm comparison. And I'm just wondering if you have any insights into why you think the Markov clustering approach worked best. I mean, is it just because it is kind of customizable through these two parameters? It has this multiple levels of customization, or could it have something to do with the process that that the Markov, essentially the Markov chain is encoding in that, the flow that it's essentially representing in the algorithm.
I think it's a combination of both. So the first one, obviously, because we have these parameters to work with, that helps a lot to customize our output. But also on the other side, I think what Louvain and Leiden are looking for in terms of a cluster by definition, and their paradigms are much different than those of information theory-based methods, which are based on random walks. Like I think they look at clustering as clusters as neighborhoods, and if I start a random walk and end up in the same neighborhood, then this should be a family of some sort. And I think that paradigm is just naturally more well suited to find clicks, because what we are doing here is a very, very basic form of clustering, because we are just trying to extract clicks. We know what we are looking for, while Louvain and Leiden are beneficial in scenarios that you don't exactly know what you're looking for, what kind of structures. It may not necessarily be clicks, it might be something that has overlaps and things like that. In that situation, I think Louvain and Leiden are still worth looking into.
And in fact, actually, we have some more recent unpublished work on clustering algorithms that are more attuned to really finding clicks, that maybe even faster than MCL, but that's kind of some current work that we are kind of following on and on. I mean, MCL works pretty well for these purposes, but I think there are even probably better algorithms that are applicable to this click identification problem. I mean, if you are really interested, you have a really good, I mean, another application of this, or I mean, let me know, because we have more ideas. Or let us know.
Yeah.
Any additional questions? Yeah, I think that's it. Thank you very much. That was a great talk. Thank you.
Well, thank you.
Thank you.
I just want to say, I mean, I'm local. If you have comments or questions, please contact me. And now, even if Ruch is not local, it's in New York now, but we are all local with Zoom, so we can all just contact us, and we're happy to follow up with.
Yep.
More.
If you have any questions regarding eyelash, I'm available via email.
Okay. Thank you very much. All right.
Thank you.
Thank you.